So welcome everyone. Thank you for attending the OTS webinar. We're excited to have our first webinar since the annual meeting. And we have a very special speaker. We have postdoctoral fellow Claudia Aloisi, who will be speaking today. And so she actually did her PhD at ETH Zurich with Shanna Storla in a very interdisciplinary lab that has a focus on toxicology. And so now she's a postdoctoral fellow in Paris. And so I will pass things on to her to give us a great talk and get things started.
Thank you, Erin. Thanks everyone for being here and for having me here. So I'm gonna share my presentation. So let me know if you see it. Should be up to, right? So, So yeah, thanks everyone. I'm Claudia. I'm indeed a EMBO postdoctoral fellow and now also Swiss Foundation fellow at the École Normale Supérieure of Paris. However, the work I will present you today dates back to my doctoral research at the ETH. And you probably already had the time to wonder what is a toddler toy doing on the first slide and what does it have to do with the detection of mutagenic DNA damage using an artificial nucleotide. So we all probably had such a toy, which is called the shape sorter, where you had to find the correctly shaped brick to pair with the correctly shaped hole. And in the fancier, newest version, the correct pairing would be rewarded with sound. Well, this is obviously an analogy for the strategy behind the detection strategy we present you today, where we took advantage of the specific geometry of a modified DNA base within a specific enzymatic context, which is here in the red box with the red molecule, to find its perfect pairing partner, the blue molecule, as a first step to develop a detection strategy. So in our analogy, the pairing partner is the brick and the damage within an enzymatic pocket is the shape. And we further developed this in a way that the mere presence of the blue molecule will signal for the damage in a way similar to the sound of the toy. And while I will present this for a specific kind of DNA damage, in principle, this strategy could be extended to any shape and to any damage. And before actually going into the specific strategy, specific damage, I would like to take a step back and give you a small overview of other brick-like strategies which have been developed in the group of Shanna over the years. So synthetic nucleosides in general have been developed by several groups for essentially for four purposes: for expanding the genetic code, for optimization of aptamers, to study the properties of polymerases, and to probe for modified bases, modified DNA bases. And it's for the two latter areas that Shanna and our group developed over the years a small array of single nucleotides. And the family of damage we were interested in is O-alkylguanine here, showed on the top figure. And we designed originally by modeling, computational modeling, we designed some probes that would pair with O-alkylguanine, as you can imagine, thanks to the capacity to form hydrogen bonding here in highlighted in blue and yellow. And over time, we expanded this small library according to the results of stability obtained with the previous partner, and also to explore different hydrogen bonding patterns, exploring different stacking interaction due to the conjugated part of the probe. And in this way, we were able to interrogate the mechanism and the importance of hydrogen bonding and stacking interaction for the functioning of polymerases, particularly TLS polymerases, translesion synthesis polymerases. And over the years, we, by testing different pairs, different polymerases, we came to the conclusion that hydrogen bonding is indeed a major driver of the efficiency of replication. This is not surprising, although there are some groups which worked with synthetic nucleotides which were not capable of forming any hydrogen bonding, and the pairing was only based on geometry complementarity, and they would work as well. But when the hydrogen bond can be formed, this is actually, at least in our hands, was the major driver. And indeed, I will not show you all the works throughout the years, but I just want to leave you with this latest work, which a colleague of mine has been doing, where he actually tested 24 permutation of base pairing, and indeed found and confirmed that the hydrogen, maybe here you have a little box, I should close it. Yeah, so the hydrogen bonding was the major driver. Of course, there were some outlier, which will account for other geometrical and interaction stacking, for example, stacking interaction properties, but more or less the major driver and the trend was that of the hydrogen bonding. The second area of interest where we used synthetic nucleoside was indeed the probing for DNA damage, DNA modification, which in our case was O-alkylguanine. And in a first moment, we utilized these synthetic nucleotides in a statistical way by just exploiting the selective hybridization of the damaged base with the artificial nucleotide. And for example, we did that by fluorescence, which I'm not showing you here, or for example, with these nanoparticles, which would aggregate in the presence of the damage, thanks to the selective hybridization, and then change color, change absorption coefficient, and change color. In the second moment, to develop detection strategy, we also introduced an enzymatic factor. So we didn't just look at the static hybridization of the two of the pair, but we put that into the context of DNA synthesis. So we had a polymerase that would synthesize DNA opposite the damage or past the damage, introducing another layer of selectivity. And I will not go too much into details because today's presentation, my PhD project, was based on one of these strategies here. So now we can ask ourselves, what was the purpose of my project? Why DNA damage detection? So as you all probably know, DNA damage is toxic and potentially mutagenic, and yet every day our cells are continuously threatened by endogenous and environmental damaging agent, causing a staggering 10,000 to 100,000 damage per day per cell. So obviously cells across species evolved response and mechanisms, which consist either in the simple DNA damage repair. In some cases, the damage can be tolerated, meaning that the metabolism of DNA will continue regardless, despite the presence of damage. Or when none of the above is possible or convenient for the cells, then cell death will be induced, obviously causing toxicity. But also DNA repair and DNA damage tolerance have dark sides, as in biology, there is never one face to the coin. And particularly DNA damage tolerance is often associated with lower fidelity of replication or transcription, which will introduce potentially errors in the replication and hence mutations. You can clearly imagine that, but just to give you visual idea of what that means, when you have your damaged DNA, from now on I will only refer to adduct, covalently bound to the DNA, and you will replicate the damaged DNA, you will likely introduce a mutation, either a point mutation or a more extended one, depending on the damage and the polymerase, obviously. When you do that, the next round of replication, you will introduce the mutation opposite the damage. I don't know if you see the mouse here, but most importantly here, the already mutated DNA will fix such mutation and potentially accumulate more, which can potentially lead to cancer-driving processes. And while this relationship is well established for some type of DNA damage and some type of cancers, this is not the case and remain elusive in some other instances. And that is the case for the damage of today's presentation, which is O6-carboxymethylguanine, DNA damage, which is formed by alkylation of DNA from nitrosamines, either by direct alkylation or via the formation of diazoacetate. Nitrosamines in turn are formed or found upon exposure and consumption of red and processed meat or cigarette smoke. And once O-6-carboxymethylguanine, from now on CMG for short, is formed, then, as we saw from the slide before, this can introduce the mispairing of a thymine, so an error, and that can result in mutation, a G2A particularly, and then accumulation of mutation. And because of the origin of CMG, and because CMG has been detected in colorectal DNA, and induces a mutation which highly resembles that of some type of cancers, CMG has been hypothesized as a cofactor in the development of colorectal cancer associated with meat consumption particularly. But there are some challenges in making that direct connection for sure. And some are gaps of knowledge. For example, what is determining the accumulation of CMG, what is repairing it, and so on and so forth. And other gaps and challenges regard the technologies. At the moment of my project, more than now there are some techniques coming up, but not so many. Especially at the moment of this project, a few years ago, there was no way to detect CMG and retain the the sequence context information. So there was no way to know the distribution of the damage across the genome, which is a very important factor to then associate the mutation, the damage with the rise of driving cancer mutations. And so this is when we came into play. We strived to develop a detection strategy for CMG, which would retain the sequence context, so that we could then look at the distribution of the damage as a future perspective, and then relate that to the mutation. So I already spoiled about what the detection is based on. Obviously, we looked at nature. We knew that we had to compete with the natural basis for finding a pairing partner, our brick for CMG. And so we looked throughout our library. We created also a few more from that library. And eventually, to make a long story short, we found that the best partner for CMG in this context of the enzymatic pairing was expensive. So indeed, normally I don't give the overview at the beginning, and so here I say that our detection strategy is made of 3 elements. And the first one is the chemical, the synthetic nucleotide. The second is the enzyme, which actively introduce the nucleotide opposite CMG. And the third aspect, so the first two aspects concern the development of the biochemistry to mark CMG. And the third aspect consists in turning these into an analytical readout, so that we can not only detect CMG, but also quantify. What about the sequence context, which I stressed very much in the background? Well, because we are not only, as I said before, we are not only hybridizing expansive with CMG, but we are pairing expansive opposite CMG during DNA replication. And for DNA replication, you need a primer. Is the primer that is what will target a specific sequence context, and so allow us to retain this information. And so the experimental setup of most of the experiments is the following. I had a plating DNA in red with or without the damage. This would be paired with a primer, and the primer would be extended of 1 or more bases, and in order to study the kinetics and the efficiency of the process. And for the highest sensitivity at the beginning, I was radiolabeling the primer. And then I was feeding the system with the nucleotides one at a time, so that I could study the single incorporation. And I didn't want to change the figure from the publication, so I hope this is not too confusing, but I will not discuss here benzi, which is another artificial nucleotide, but only expenzi, and compare that with the thymine. So I hope this is not too confusing. Let's not look at benzi here for the moment. And then, so after feeding the system with nucleotides and incorporating the single nucleotide, I will look at the efficiency of the incorporation through a gel. The bottom line of the gel is always the primer, which is radiolabeled, and anything above that means that the primer is longer. So it runs above, it runs lower. And so here you have only the +1 base. In the case of G, so unmodified DNA, it's not very interesting. But in the case of CMG, you see how expensi is highly incorporated opposite CMG and significantly higher than the thymine, which we know it's, and I also tested, is significantly higher incorporated than the cytosine, the natural base. By performing this at increasing concentration of all the nucleotides, I was able to measure the kinetics via Michaelis-Menten model. And I will not show you the full table, but just the most important pairs. So by doing the Michaelis-Menten kinetics, I could get the catalytic efficiency, so Kcat over Km, and compare among base pairs. And you can see here how expensi, triphosphate, is paired opposite CMG 70 times more efficiently than opposite G, which is good for the selectivity, and 7 times more efficiently than the natural T. To put this into structural context, I performed some molecular modeling, and so within the enzymatic context by modifying a previously published crystal structure of the enzyme. And so what I found is that indeed expensi triphosphate opposite CMG, as you imagine, forms 2 hydrogen bonds, while expensi opposite G forms only 1. And on top of that, there is a steric hindrance between these 2 hydrogen here, which in turn will tilt slightly the expensi, and so the resulting base pair expensi G will not be perfectly planar like an ideal base pair. Meanwhile, expensi CMG will really highly resemble the natural base pairing. When we look at the thymine, so compare the T with expensi, we see that the situation is not very different in terms of planarity and hydrogen bonding. But as you remember, maybe I didn't mention enough, by designing these artificial nucleotides, we also introduced some P surfaces which could interact either with the neighboring bases in the case of the isolated hybridization or with amino acids from the enzyme. And so we do expect the difference between expensi and the thymine to actually lies on the interaction with the surrounding enzyme. And indeed, when we look at the interaction of expensi in the enzymatic context while pairing opposite CMG, we see that this is at lower energy, so more stable than the one of the thymine. So having studied that, we were ready to move on to the next step. So we now know that expensi is favored opposite CMG compared to the other bases. So now we want to know whether expensi is able to recover the phenotype of DNA synthesis past the damage. So whether the DNA can be synthesized in the presence of the damage. And so the experimental setup is the same, but this time I feed the system with all the nucleotides plus or minus expensi. And so, as always, G is not so interesting. It's just good to know that it makes sense. So you have a full extension and complete synthesis in all cases. But in the case of CMG, as you see, when you don't provide expensi, there is very low full synthesis product. However, when you introduce expensi, this increases of 4-folds more or less. So Xbenzi indeed recovers the full extension of DNA synthesis past CMG. And so we are ready to take the step forward, the quantification, the third part of our detection strategy. This time I basically repeat the DNA synthesis, but without radioactivity, because I needed to further process the sample. And of course, we didn't have everything dedicated to radioactivity. So I would perform the extension of the primer to the full length several times by using basically the same template over and over again, so that I could somehow amplify the signal for CMG in a way that for one molecule of CMG, I would get more molecules of Xbenzi. Then I would digest my DNA sample to the nucleoside level. A technician, mass spec technician in the group, developed a method for the detection of Xbenzi nucleoside. And then by performing these at increasing concentration of CMG, I was able to get a linear relationship between the detected amount of Xbenzi and the original initial amount of CMG. This is more or less where I left the project at the end of my PhD. So we successfully designed and synthesized a pairing partner for CMG, which would allow and promote the full length application past CMG, and which would mark for the initial amount of CMG, as well as the location, which is intrinsic in the sequence of the primer. But as I mentioned, now I am in Paris. I will not, this is out of the scope of today's presentation, but just to give you an overview of what I'm doing, and in case you're interested, you can contact me further. So I am now indeed in Paris. I have a chemistry-based education. As Erin said, I did my PhD in Shana Sturla group, which is highly interdisciplinary, and that was indeed the purpose of me choosing this amazing lab, where I could indeed apply my chemistry knowledge to a more biological setup. So the project I presented you today was chemistry, biochemistry based, but I also performed some molecular biology projects, looked at the repair, for example, CMG. And because my vision for my research is that I want to use my basic science or chemistry knowledge to study biological processes, which are obviously more complex and more multi-factor, multi-step processes at the molecular level. So after indeed my PhD and being exposed to more biological and biochemical setup, I came across single molecule strategies, and I love the way how they allow really to look at the molecular level, which was my original vision when I studied chemistry for my bachelor. And so this is why I now joined the group of Terence Strick in Paris, where I do single molecular strategies to study DNA repair. And again, I will not talk about the project. I just want to say that there are two amazing strategies to look at the binding of proteins and the kinetics of repair, namely total internal reflection fluorescence and magnetic trapping, which allow me to gain this very complementary information. And in case you're interested to know more about that, you can reach me here. And this brings me to the end of my presentation. I want to thank you all for being here, for your attention. Of course, Ronald and Erin for coordinating or organizing all of this, the OTS for the possibility to be here, and then funding group where I am now, group where I've been, it has been a blast so far. And thank you so much.
Great, that was amazing. It's always really cool to see what's going on in the space of modified oligonucleotides, and especially in their amplification, because that's one of the biggest challenges of actually incorporating them in for use, right? Exactly. So we have some questions. Yes. So just a reminder to participants that you can use either the Q&A function for your questions, or you can type them in the chat. And so our first question is, O6-CMG is known to be present in, for example, colon cancer cells. What kind of other biological models could you use to study sorts of modifications?
So I guess with a more biological model, I don't know if it's Ronald who wrote it or on behalf of someone else, is that I guess like the system where they study, because in this moment I was just using oligonucleotides. So the idea indeed was to and is to go to cells and eventually also in vivo. However, the sensitivity at the moment is not quite there to move there. We are able, with this strategy and similar strategies in the group, which we've been developing since I left, we are able to look at CMG when we spike, when we treat the cells with the carboxymethylating agent successfully, but the level of O6 CMG in vivo samples or even in physiological levels is very, very low. So indeed, I mean, that is the goal, the final goal, but for the moment, it's kind of a proof of concept. We can do that, and we need to improve the sensitivity, sorry.
Yeah, so my question, I think that's part of the answer. It was my question. So, but because it's always, you know, you want to have to study this in a more like physiological situation,
yes.
right? So preferably,
yes.
well, I would say the first step would be like a very good cell culture model where there's a lot of O6 CMG. But
yeah,
if that's not there, then it's difficult to study, right? So that might
exactly. It's extremely low. Even methyl G, which occurs at a higher level, and the nanoparticle project was developed for methyl G. They had to spike the cell because the sensitivity is not bad. It's comparable to mass spec and other techniques, but because you only look at the specific sequence, you're even reducing more the level and the pool from which you're looking at. So you're not looking at the overall amount, but just in that sequence. So that makes it challenging, and for sure the cell model would be is the final goal. And I mean, now we're really moving towards more damage mapping techniques, which would allow to go move towards there. But yeah, definitely.
Yeah, it might be. I think so. You are working on like a single, on a very nano level, right? So, but also I think also the techniques that are currently being developed to study like single cells or even in more detail are also developed now. So that might also help in studying the effect.
Exactly. Yeah.
And so are there people known that make these kind of models?
Sorry, people know?
Are there models known? Are there people? Are there labs out there that make more of these kind of models?
You mean for CMG or in general? No, for CMG, not really. I mean, even for the, as I mentioned, I was studying the repair of CMG. There are so few works about it, and because it is, so there were studies regarding the exposure to meat and consumption of meat a few decades ago. And then it kind of was dropped there. The attention was moved towards more abundant DNA damage as regards the detection. So there isn't so much out there, and there aren't models, as far as I know. So even you mentioned like a good model for cell model, there isn't any. So we would need to create one or spike one or. Not as far as I know. Obviously, I left the group a few years ago, and I'm still in touch by mentoring a few people, and also just in touch as friends. But I'm not. It's not my main topic anymore. But as far as I know, there isn't a model for that.
Great. So I noticed in kind of talking back and forth and researching you in preparation for the webinar series, that you've actually won some pretty prestigious awards. And so I don't want to put you on the spot, but just to kind of like shift focus a little bit, because we have a lot of trainees that attend our session. I'm just wondering if you have any advice specifically for trainees in terms of applying for these types of awards or finding a good postdoc, or just any kind of general advice, even if it was something that someone told you that you found helpful, just to kind of close off the session if we have no more questions.
Yeah, absolutely. So indeed, I got this award, which gave me an amazing opportunity to tour around Switzerland, because it was a national prize, and to meet individually one-to-one with many people. And one professor, indeed told me, he called this kind of awards beauty contest. And he told me that in the US or in the North America, there are more of these. And that obviously it didn't mean it in a mean way that is useless, but it's more like a way to get visibility and exposure and so on and so forth. And so he told me that it's an essential part of building up your career towards academia. He was trained in the US, so he got many of these beauty contacts, and now he's a very successful professor. So definitely this is a very good opportunity, not only for the line on the CV, but also for what it entails, because I toured around, I met so many people, I made such a big connection and network. And then in terms of career development, I mean, I'm still a postdoc, so I'm not quite there that I can give advice of success, but definitely also applying for fellowship is an important factor, makes you stand up for the next step of job search, and also gives you some independency, which I'm appreciating as a postdoc. So definitely, yes, applying for fellowship awards, as much as, you know, depending on the personality, I felt kind of shy to apply for awards and stuff like that, but it was highly encouraged from my PI Shana, and that was definitely a good thing. So yes, go try as much as you can, get visibility, get exposure, and I would say it obviously depends on what is your goal. If you want to stay in academia, do beauty contests, do apply for fellowships, because it's a good opportunity both to build up your career, but also to show that you come up with good ideas, that you get funding for those. However, if your final goal is not to stay in academia, then you take a different path, and you maybe don't need as many as of these awards and beauty contests. And maybe you want to focus more on developing some skills rather than getting this exposure.
Great, thank you. That's great advice.
Thank you for the question.
It also reminds me that the OTS has several awards to apply for scientists at all levels. So trainees up to kind of full career stage scientists, and for multiple things. So if you're interested in science communication, we have that all the way to paper of the year. So there's all sorts of opportunities for OTS to support people as well with these prestigious awards. And so with that, we want to thank you so much, Claudia. That was an amazing talk.
Thank you, really, for inviting
really
me.
well presented, very clear. And so we loved having you. Hopefully we can get you back soon. And then I'll just
with pleasure.
Wonderful. I'll just pass things over to Ronald, and he's going to tell us a little bit more about the webinars that are coming up towards the end of the year. Great, thank you.
Thank you, everyone.
Yeah, thank you very much, Claudia. So I just want to remind you that we have 2 webinars in the coming few weeks. So the first one is from Virginia Aragafala, and she will talk about the preclinical development of an approved exon skip drugs. So it will be like a story from the first preclinical development all the way to the first clinical trials. So I think very interesting story to hear. And the second one will be of an old OTS award winner. So that's Bruno Godinho, and he will talk about delivery and PK/PD of novel CNS ASOs. So I think so Virginia will talk on November the 3rd, and Bruno will talk December the 1st. So write this down in your agenda, and obviously we will send emails around on the OTS website to promote these webinars. So thank you all for listening to this webinar, and see you all next week for Virginia's presentation. Bye-bye.